Diffusion can also occur through cell membranes. However, cell membranes are selectively permeable. This means that some substances can diffuse through a cell membrane while others cannot. The flow of ions or molecules through a cell membrane as a result of diffusion is referred to as passive transport. It is referred to as passive transport as it requires no energy from the cell in order to carry a substance across the membrane. In the process of passive transport, the membrane acts as a filter. Phospholipids and protein channels control which molecules can cross the membrane and may influence the rate and timing of the movement of ions and molecules across the membrane. But they do not influence the direction of movement. Substances continue to move only from areas of high concentration to areas of low concentration. Let's now look at three types of passive transport that occur as a result of diffusion: simple diffusion, facilitated diffusion, and osmosis. Simple diffusion is the process by which gases, such as oxygen and carbon dioxide, dissolve in water, and phospholipid-soluble molecules, such as ethyl alcohol and vitamin A, diffuse across the phospholipid bilayer. This is referred to as simple diffusion because these substances can cross anywhere along the phospholipid bilayer, while most other types of diffusion, such as facilitated diffusion, which we will look at in a moment, requires a substance to cross the membrane through a protein channel or with the help of a carrier protein. Generally, the rate of simple diffusion of a substance across a membrane is a function of the relative concentration of the substance on either side of the membrane, the size of the substance's molecules, and on that substance's lipid solubility. In the process of facilitated diffusion, either channel proteins or carrier proteins facilitate the crossing of a cellular membrane by water-soluble substances such as sodium, potassium, and calcium ions, amino acids, and monosaccharides, which cannot typically diffuse through the phospholipid bilayer. As we saw earlier, each type of channel protein is typically lined with specific amino acids and has a unique diameter in order to allow only certain ions or other substances to pass. Channel proteins also often have gates which open and close in response to electrical or chemical signals. Carrier proteins, as we saw earlier, attach to specific molecules in the cytoplasm or extracellular fluid. The attachment triggers changes in the shape of the carrier protein that allows the molecule to pass through the protein and thus cross the plasma membrane. Some carrier proteins don't use cellular energy and only allow ions and molecules through the membrane that would normally pass through as a result of diffusion. However, as we will see later, there are carrier proteins that use cellular energy to carry substances against the flow created by diffusion.
This is a cell that secretes large molecules that are unable to pass through the lipid bilayer of the plasma membrane. They leave the cell instead by a process known as exocitosis. After production in a system of membranes called the endoplasmic reticulum, the molecules are packaged in a small sac of membrane called a vesicle. The vesicle moves to a stack of membranes called the Golgi apparatus. Their membranes merge, and the vesicle releases its contents for modification. As the molecules leave the Golgi apparatus, they are again packaged in a vesicle, which moves to the plasma membrane. As membranes merge again, the contents leave the cell without actually crossing the plasma membrane. This process can also occur in reverse, allowing large molecules to enter a cell. This is termed endocitosis. Sodium inside the cell binds to the protein. The addition of a phosphate group from ATP changes the shape of the protein, and the sodium is expelled. Now potassium binds to the protein. The phosphate is released, and as the protein returns to its former shape, potassium is moved into the cell. Sodium can once again bind to the protein, and the process repeats as long as there is a supply of ATP.
In small water animals, the blood carries excess water to specialized cells where it is collected and discharged through tubes back into the environment. In mammals, kidneys perform this function, keeping the water content of their body fluids in balance with their tissue cells. But suppose the concentration of salt becomes higher outside of an animal or protozoan cell. This is exactly what happens when a paramecium is carried downstream to the sea, an event we can simulate by adding a drop of salt water. As the paramecia encounter the higher concentration of salt in the sea water, they lose water through osmosis, shrivel, and die. Drink enough seawater, and the same will happen to your cells. So, osmosis can cause a decrease or an increase in a cell's water content. Red cell ghosts produced by osmotic rupture have provided biologists with pure samples of plasma membrane for use in studying its structure and chemistry. An electron micrograph of a section through the plasma membrane shows that it is composed of a double layer of molecules. Biochemists picture them like this, a double layer of fat molecules. The question is, how do water molecules get through this fatty membrane? The accepted theory is that water molecules are able to shoot through gaps or pores, irregularly distributed in the membrane. But this does not explain how other molecules cross the membrane barrier into a cell. The mechanism puzzled cell biologists until the discovery of proteins embedded in the membrane. The protein acts as a lock. When a passing molecule fits it like a key, the lock opens, sending the molecule through into the cytoplasm. This is called facilitated diffusion.